Hello and welcome back to the lecture on Chapter 3, Structure and Function of Cells. We finished up with Part 3, and we were talking about movement of materials through the cell membrane. And now we're going to talk about, now we're all the way on the inside of the cell, and we're going to talk a little bit about the cytoplasm of bacteria, but mostly we're going to talk about something that some bacteria can form. All right, so let's talk about cytoplasm. So cyto, remember, cell, and cytosol, we're talking about the liquid portion of the cytoplasm. So cytoplasm is all that stuff that's on the inside of the cell. Cytosol is mostly water. And in this space, or in this area, there is the cell's, genetic information. If we're talking about prokaryotic cells, remember that there's not a separate nucleus, there's not a membrane around all the DNA, but wherever the DNA is referred to as the nucleoid. So it's like the kernel-like area. And it just doesn't have a so it doesn't have a plasma membrane around it. But there are areas, inclusions in the cell. So there's areas where we have reserve deposits of chemicals. Think like storage, but I don't have a storage compartment to put it. So I just have a little pile of these molecules. So here's something we'll get to again later, especially during the genetics section of the class. Remember the pilus? This is one bacterium that can transfer some of its genetic information to the cytoplasm of a different bacterium. Cool stuff. All right, continuing with this part of the chapter, we go over a section called endospores. So endo, within, and spores is like a little solid thing. This is actually the secret to certain bacteria being so resistant to cleaning and being able to get rid of them. It's super stable, dormant defensive cells. So when the bacterium is in this form, it's not actually metabolically very active. So it actually can survive for a long time, like years, in the soil, for example, or just in hospital bedding. It's really hard to get rid of some of these things. Now, what's fascinating is it's only when the environmental conditions start to turn bad that the bacteria will resort to forming these endospores. But man, when they do that, they're just hunkered down and they can survive so much. And not every bacterium can do this. The kind of famous ones for it are the Clostridium bacteria. So Clostridium difficile, you may have heard of it as C. diff, if you've ever heard about some of the bacterial infections that lead to the severe diarrhea, especially in hospitalized patients. There's Clostridium tetani. This is the C. tetani. This is the one that you have to get a tetanus injection to protect you from. And if you associate that with rusty nails, I'll explain why. We're always talk about, rusty nails in the ground and, somebody steps on it and then they, we're afraid they're going to get tetanus. Here's the thing. It's not the rust on the nail. It's the fact that it was in the soil and then it can break the skin. The Clostridium in the soil can survive for years and years and only gets activated once it gets into somebody's foot. And then once it's in somebody's foot, wow, that's a party, because there's all these resources available and then the thing starts reproducing like crazy. And it produces a toxin that can cause contraction of the muscles. Okay, and then Clostridium botulinum can do the opposite thing. C. botulinum, this is what botulism is. Sometimes there can be canned foods that get broken open or just a hole in the can and some food goes bad in there. It's this kind of the Clostridium botulinum can lead to this problem. Its toxin causes paralysis. And if you've ever heard of Botox, this is what we're talking about, is the botulism toxin. People inject that into their forehead so to get rid of the wrinkles on the forehead. Yeah. And there it's cosmetic, but imagine that throughout the body and especially affecting the diaphragm. Paralyzed muscle, can't breathe, that can be deadly. All right, so the point of this, what do they all have in common? They all belong to this group, the Clostridium bacteria. And one of the things that they all have in common is that they can form these super stable endospores. And Bacillus cereus. Let's see, this one can also cause food poisoning. It's just hard to clean, hard to get out of, like industrial places. And so it's a defensive strategy that the bacteria have to survive unfavorable conditions. The vegetative cells that can transform into the endospore. So it's like, I'm going down. Like if the conditions continue on this trajectory, we're going to die anyway. And so they kind of hunker down and make this very tough structure that can survive really harsh environmental conditions. So resistant to extreme conditions such as heat, radiation, chemicals, you know, all the things that we do to try to clean surgical instruments or, you know, yeah, clean industrial food areas. They're a problem. Here's a cartoon kind of detailing what happens. The cell starts experiencing difficult conditions, and then it starts to kind of, I'm putting my resources into surviving the next few months or years. And it kind of hunkers down and you just get this super thick outer layer. There's cell membrane, there's peptidoglycan, there's the spore coat, there's another outer coat, and then this thing, almost indestructible. And then we wonder, like, well, why can't we get the hospitals clean enough that the patients don't keep coming down with C. diff? It's because of this. It's because of this structure right here. It's almost impossible to get rid of. And here's a question for you about endospores. Which of the following statements is true? A, endospores are formed as a way for prokaryotic cells to reproduce. B, endospores can be easily destroyed with household bleach. C, endospores form in response to harsh environmental conditions. Or D, endospores do not survive in the environment for more than a few days. And I think we can eliminate that last one for sure. Not easily destroyed. It's not generally a form of reproduction, but rather like survival. And the endospores start to form when the cells start to experience damage from harsh environmental conditions. They know to hunker down, or they know, I don't know. The reaction to the harsh conditions is this very tough endospore, and you can think the evolutionary history of this. You know, those bacteria who didn't do that would die, and those bacteria who did do that would survive these harsh environmental conditions to reproduce on a better day. And then finally, this is going back to something we saw earlier in the chapter. A bacterial cell has a cytoplasmic solute concentration equal to 0.85% sodium chloride. So almost 1% sodium chloride is placed into a tube containing a solution that has a concentration of 0.2%. If you get yourself trying to like compare this, think just round, go, okay, 1% sodium chloride into less than 0.5% sodium chloride. Is that 0.5%, is it more concentrated or less concentrated than the cell is? Do the math, it's less concentrated. And what's the term that we use to mean less concentrated than the cell? It's going to be either hypo or hyper. If it's less concentrated than the focal cell that you're interested in, then we say hypotonic. So this kind of solution. If the cell has been placed into this dilute solution, that's a hypotonic solution. All right, and that is the end of chapter 3. Hope you have a great week.